how many people in the NDT program? So you guys had a lot of work to do. Was it rough?
Yeah.
At times. It is rough. I think that first summer in NDT is very tough because it's an area you've never dealt with before. You're talking about waveforms, you're talking about things you've never thought about before. It's only when you go into clinicals that stuff begins to make more sense. But just persevere. That's the best thing I can tell you. Persevere, because sooner or later everything starts to make sense. But it takes time. It takes time. Now, we're one of the few classes that are meeting in the building. Most classes this semester are being conducted online. Okay? Even with this class and some of the clinical classes, once Thanksgiving comes around, we're going to transition to online teaching. And for that reason, primarily for that reason, I introduced you to the lockdown browser. So what I'm going to do right now is simply go through a syllabus. There's some changes, and I want everyone to become familiar with the syllabus. All right, the beginning here simply is a description of the course and the rationale. This is material that you've seen in previous syllabi. Okay. So requirements for the course, sir. Okay, part of the course is going to be online toward the end. So you need to have a computer, a laptop, or a mobile device, and you have to have access to a webcam. Camera. Okay? You need to have a stable internet connection. Okay? If you don't have these things here that are listed here, then you can't take this course. This is a requirement for the course. Now, if you don't have them and you want to take the course, then we need to have you hook up with IT. IT does have a few laptops that are available to students who can't afford or don't have a webcam or whatever. You can check with IT. Okay. This part here was required by administration. Any exams or quizzes given online will be proctored, and it is expected that students will have his or her video turned on during the entire testing period. During the exam, the student may not leave the room without permission of the instructor and cannot communicate with anyone else during the test. Okay? The video display must be positioned in such a manner as to show the student's face. Those of you who tried the lockdown browser are familiar with the fact that you're being proctored during the exam. Okay? And what it will do is it will capture and report any abnormalities. Okay? One of the things that we report on a few students is that when you show your surroundings, you need to show not only what's on the walls around you, but you have to show the desktop all around you. Second thing, the light has to be proper. You have to have good lighting in the room in which you're taking the test. If you need to focus on a computer screen throughout the test. If it sees you diverting your head to one side for a prolonged period of time, it will capture that image. Okay? And that will be reported as an abnormality. If you stand up and leave the visual acquisition region here, okay? So it sees you're moving away from your seated position, it will capture that image. Okay? And it will report any discrepancies. And so, make sure that you follow all. If you talk during the exam, if someone else enters into the room, it will capture that. Okay? So make sure you follow all the rules. It has to be able to see your face at all times.
Dr. Fenner.
Yes.
With Kylie doing virtual school as well, she'll be sitting at the desk with me. We're normally sitting at the kitchen table. And it says no other electronics.
I want you to
send
that email to, send an email reporting your problem, and I'll send it up to administration, okay?
Because she has to be online between 8 a.m. to 3 every day.
And you have to be in the same room right next to each other?
We don't have to be in the same room, but I have to monitor her.
Send that to me.
I'll
Because,
send you the paperwork for the online, the
Send-
virtual learning
no,
requirements.
send me the information about what your requirements are. Because I have to forward those to administration. You can't have a dog in the room with you, or a pet, while you're taking the test. Okay, so if all of a sudden you're taking the test and a dog comes into the room, it will capture that. Okay? These are the same rules, by the way, that are applied when you go to a testing center. If you want to apply, for instance, to PT school or to OT school, or some graduate program like PA school, and you go to a testing center, okay, the same rules apply. Okay, you can't be talking, you can't bring a pet in there, you can't bring a child in there. These are the same rules, or else they will negate the test. Okay.
I have a question.
Yes,
What if dogs are barking in the background?
it'll capture that.
Okay, I have 6 dogs, so
Do your best to keep them quiet. That's all I can say, otherwise it's gonna capture and put you, it's gonna, let me say this. I can go back, I can request the video
okay.
and go back and require an explanation for each of the, but to the best that you can, keep the dogs in a separate room.
Okay.
And the truth is if they're making distant barking sounds, it's not gonna pick that up. Okay? You can, just keep them in a room at the back. Like for instance, if we hear some lightning, it's not gonna pick that up. Okay? It's gonna pick up close range activity. All right, so this year, and it's for this year only, they have subdivided the course into lecture and lab. Now, I'll tell you what happened. Previously, this was combined lecture lab course, but administration came in a department meeting and they say all science classes have to be subdivided into lecture and lab components. So that's why this is subdivided into a lecture and lab. The truth is, I did research for all other universities in this region, and with the exception of one other university, nobody subdivides the histology into lecture and lab. It's always combined. And that's because histology by nature is a laboratory course. Okay? So for this year only, there's going to be a fall separation into lecture and lab. Next year, we're going back, because I reported what I found, and so administration said, okay, that doesn't make sense then. But it was too late. So this year, we're kind of breaking it down into 2 components. So the lecture is going to consist of, the grading for the lecture is going to consist of 5-unit tests. Okay? That counts for 100%. There are no dropped exams. Okay? What happens if you miss an exam without a valid excuse? Well, you simply get a 0 for that exam. A valid excuse would consist like a doctor's note, from the emergency room, or you have a jury duty summons. Okay? If we have to give you a makeup, let's say for exam 2, it's going to occur at the back end of the semester. And the makeup will be different. It may be a essay type test, but it's not going to be the same exam that other students took. Okay? So it may be, the format may be different from the original exam. This is the grading scale, you're familiar with this. Okay. Attendance is important. If you're not present at the start of class, you'll be marked absent. Okay? Let's say you come in 5 or 10 minutes late, you're tardy. It's up to you to come in during the break between the lecture and lab and say, would you convert that absent notation to tardy? If you don't, it's your responsibility. Then you go on as being marked absent. Okay? If for any reason you miss 30 minutes or more of class, that counts as absent. Now I'm glad to have you here that remaining time, but you have to be here throughout most of the course. Part of the course may be online, conducted through Zoom, for instance. Okay? By enrolling in this course, you agree to show your face on camera throughout each class session and participate as appropriate. We had some meetings after last semester about the experience of being online. And the teachers all agreed, and so did administration, that participating in Zoom sessions means that you have to have your video on. Just like in a regular class where the teacher can see you and interact. Some of the teachers complained that during the course of the Zoom session, students turned their video off, so they couldn't tell if they were in attendance or not. You can't do that. This is administration requested we put this in the syllabus. And if you can't agree to this, let us know up front. And we may say, well, this might not be the best course for you. Logging into the Zoom session doesn't guarantee you'll be marked present. Okay? You have to log into the Zoom session and be present the entire time. Now, if you have to leave for some reason, ask to be excused, and hopefully you'll come back as soon as you take care of whatever your needs are. Class etiquette. Okay? Civil behavior is expected by all members of the class. And this is just common sense stuff here. If you have a disability for which you need accommodations, you need to contact Meredith Reed. Okay? No cell phone usage during class. No recording of the class. This goes through academic honesty. And here's your syllabus. Now, this syllabus is going to change already, simply because of the storms. So, for instance, we're already a day behind. Also, you can't start doing lab until we have a few lectures. You know, you can't just start looking at tissue without any prior instruction. So today we're going to spend both sessions with instruction, with lecture. And then what you'll find is that we'll use perhaps the early session sometimes for lab, the later session for lecture. It may vary. Does anybody have any questions? Okay, let's see. So today we're going to talk about tissues. Back to A&P 1. The first lecture you ever had in A&P 1, we talked about the fact that molecules form bonds that give rise to structures. And the smallest structure of life, we said, was a cell. We went on to say that cells will link up to form tissues. Okay? In turn, tissues will link up with each other to form organs. Organs will link to form organ systems. Okay? And all the organ systems together make up the organism. So our focus this semester is going to be on tissues. You may recall that there are 4 primary tissue types: epithelium, connective tissue, muscle tissue, and nervous tissue. So we're going to take these in sequence throughout the semester. We're also going to discuss how tissues come together to form organs. I'm going to look at organs under the microscope. Now, these primary tissues, epithelium, connective tissue, muscle tissue, and nervous tissue, are derived from primitive embryonic germ layers. Okay? These germ layers are formed during the first few weeks of life. Right? These germ layers are referred to as endoderm, mesoderm, and ectoderm. So these early germ layers, these poorly defined tissues or groups of cells, will give rise to these 4 different primary tissue types. We're going to talk about how these primary germ layers are formed and how these give rise to tissues. But we're going to start where we ended in A&P 2. The last unit we discussed in A&P 2 was what? Pregnancy and reproduction. Okay? And we talked about how a spermatozoa fertilizes an oocyte. we said that when the spermatozoa approaches the egg, it releases acrosomal enzymes. And these enzymes break down the barriers that surround the oocyte. The enzymes break down, for instance, this group of cells called the corona radiata. And they break down the zona pellucida, this glycoprotein membrane. Okay? So that the fortuitous spermatozoa that comes along at the right time has an opening in which it can contact the cell membrane of the oocyte. So the early spermatocytes simply break down those barriers. The one that comes along at the right time where there's now an opening to the membrane is the one that's going to perform fertilization. And when that first spermatozoa does that, touches the oocyte cell membrane, its head gets incorporated into the oocyte. The tail is detached. Okay? And that head contains the chromosomes of the spermatozoa, the 23 male chromosomes. Okay, the 23 chromosomes coming from the spermatozoa. Okay. Point. So what happens next is, okay. So we see the detached sperm tail. Here's the head of the sperm containing the chromosomes from the spermatozoa. This is now referred to as the male pronucleus. Whereas the nucleus of the oocyte is now referred to as the female pronucleus. And they do a little dance and begin to approach each other. Okay, these are two different packets of 23 chromosomes. And as they approach each other, their nuclear membrane breaks down. It begins to disintegrate. And so they come closer and closer to a point where the chromosomes then join together. So now for the first time we have a single-celled individual with 46 chromosomes. This is referred to as a diploid number of chromosomes. And as soon as this occurs, you can see a spindle is beginning to form, indicating the beginning of the first mitotic division. Remember, mitosis is cell division, in which each of the daughter cells receives a full complement of chromosomes. And so you see this zygote undergoing cell division, finally dividing into two different cells, still surrounded by the original zona pellucida. This type of cell division that occurs early on, following fertilization, is called cleavage. And the daughter cells are referred to as blastomeres. This first cell division takes about 36 hours. Now, these daughter cells are going to go on to divide themselves, and their daughter cells are going to divide. But each of the subsequent cell divisions are going to proceed much more rapidly. It's simply that first cell division that takes a long time. So here we see the structure with two cells. These two cells begin to undergo mitosis. Now we see a four-cell structure. These cells undergo mitosis. So we begin to see a ball of cells. Now, once you have a ball of cells that numbers at least, let's say, 16, the structure is referred to as a marula. Marula. So this process of cell division is occurring as the fertilized egg is moving from the distal end of the fallopian tube toward the uterus. It's maturing. Now, the next step is for a little hollow cavity filled with fluid to appear within the marula. You can see here you have a ball of cells, and here's that little hollow cavity filled with fluid. That's called a blastocyst cavity. Blastocyst cavity. You can also call this a blastocele. The entire structure is called a blastocyst. It's at this stage when the preembryo, we call this a preembryo, is first entering into the uterine cavity. Okay. So let's go through the terms. The pre-embryonic period is those first 2 weeks following fertilization. So during this pre-embryonic period, for instance, a marula is formed and then it gets converted into a blastocyst, and that blastocyst begins to implant. The embryonic period begins with the start of week 3 and continues to the end of week 8. So from weeks 3 to week 8, that characterizes the embryonic period. This is the period, the embryonic period, when those primary germ layers, the ectoderm, the mesoderm, and the endoderm form and begin to give rise to the 4 tissue types. In fact, this process, where the germ layers give rise to epithelium and muscle, nervous tissue and such, this is called histogenesis, which simply means the genesis of tissue. And then as soon as the tissue is formed, it begins to link up in such a way as to form organs. Okay. And this occurs early on during pregnancy. This is called organogenesis. So all of this starts during the embryonic period. The fetal period is going to extend from week 9 until birth. So those are your definitions. The embryonic period is the most critical period of gestation because this is the time when all the major tissues and organs are forming. This is the time when a pregnancy is most vulnerable, okay, to the injurious effects of environmental agents, medications, etc. Okay, so you probably remember that fertilization occurs within the ampulla, the distal third of the fallopian tube. Okay. And so here, as the structure moves toward the uterus, here we have a fertilized egg, formed a zygote, and it's beginning to divide. So that near the proximal end of the tube, you see a marula has formed, a ball of cells. As it's entering into the uterine cavity, it's given rise to a blastocyst. Okay? This is around day 4 or 5. Now, the blastocyst is going to move down toward the cervix until it finds a region where the endometrium is ripe, is ready to receive it. If it's ripe early on, it will implant higher up. If it's not ripe or ready, it will keep moving down toward the cervix and implant further down. Now, remember we said a blastocyst is a ball of cells. But as pregnancy proceeds, this ball of cells changes shape. So at one point it's fairly spherical, but with time it begins to elongate. Okay. And take on a 3-dimensional shape. That's typical of what you think of when you think of an embryo. Okay. But if we were to take this early blastocyst, let's go back. As that cavity forms, here's the blastocyst cavity. Okay? The cells get mobilized to one side. And we see that the cells begin to differentiate into 2 groups that they've color-coded for you. You have this inner group of cells, that's called the inner cell mass, and you have the layer of cells that remain along the periphery of the structure. That's referred to as the trophoblast. So with the development of the cavity, the ball of cells now begin to differentiate into 2 groups. The inner cell mass is going to give rise to the embryo. The cells that make up the trophoblast are going to give rise to the placenta. Okay. So here's your blastocyst cavity right here. You're going to have kind of an inner group of cells here that are making up your inner cell mass, so you can call it an embryoblast. And then the cells that remain along the periphery here are your trophoblast. Now, implantation tends to begin at the end of about 1 week following fertilization. So here at day 8, you see the blastocyst cavity beginning to penetrate into the endometrium. Okay? The inner cell mass, by this point, has differentiated into 2 layers that they're color-coded for you. Okay? The layer that's closest to the endometrium is called the epiblast. The layer that's further away is called the hypoblast. Here's the blastocyst cavity. Notice that your trophoblast has also begun to differentiate into 2 groups of cells. The cells that continue to form the periphery of the structure, okay, those cells are referred to as the cytotrophoblast. But then you have another group of cells that have begun to penetrate into the endometrium. Those cells are called the Syncitiotrophoblast. The term syncytio comes from syncytium. A syncytium is a group of cells for which the cell membranes between have broken down. Okay? And so what you really have is a multinucleated tissue here, where cell membranes have broken down, and this tissue is releasing digestive enzymes. The digestive enzymes are digesting away the endometrium and allowing the blastocyst to penetrate deeper. So again, to repeat, the inner cell mass is subdivided into an epiblast and hypoblast. The trophoblast has subdivided into 2 groups of cells, one of which is the cytotrophoblast, the other is the syncytiotrophoblast. Again, the trophoblast is going to give rise to the placenta, or at least the embryonic part of the placenta. The inner cell mass is going to give rise to the embryo proper. So, if we take this a day further into day 9, what you see is that the cells of the inner cell mass, the epiblast and the hypoblast, have begun to divide. In fact, the epiblast, these cells have divided and split, okay, forming a cavity between. This is the amniotic cavity, and the cells form what's called the amnion, which is one of the extra-embryonic membranes. The cells that were part of the hypoblast are dividing, okay? And they're beginning to surround the blastocyst. They're giving rise to the yolk sac. And all the while, the blastocyst is moving deeper into the uterine wall. Okay, so again, epiblast is giving rise to the amnion. Hypoblast is giving rise to the yolk sac. And in the meantime, the parts of the trophoblast are burrowing further into the endometrium. This is day 14 at the end of 2 weeks following fertilization. Notice that the yolk sac is completely formed. The amniotic cavity is formed. And you see this extension forming, this connecting stalk to the trophoblast. Okay? This is going to eventually form the umbilical cord. Okay? Notice that here's part of the trophoblast here, and it's forming these little finger-like projections called villi. I want you to remember that this structure, by this point, is not spherical. It's beginning to elongate. And we're just taking a cross-section through it. What we've done here, the color coding's the same. This is we're just showing a part of the what's called a bilaminar disc. Okay, let's go back. This structure with the epiblast and hypoblast is called a bilaminar disc. What we're going to do is take the roof off of this and look down from the surface. Okay? If we do that, we see the surface of the epiblast, and deep to it, remember, is the hypoblast. Okay. The next stage is we're going to move from a bilaminar disc into what's called a trilaminar disc. Another name for that is a gastrula. And so this process is called gastrulation. So what happens during gastrulation is that cells from the epiblast begin to migrate toward the center and form a little streak here, a linear streak, through which the cells invaginate, move deeper. And as these cells multiply and move deeper, they begin to detach. And some of the cells then incorporate themselves into what used to be the hypoblast. But these new cells that are now situated along what used to be the hypoblast become known as the endoderm. Okay? In the meantime, cells continue to detach, okay, and position themselves between these two original layers. They're color-coded red. These cells that are going to be positioned between, sandwiched between what used to be the epiblast and hypoblast, become known as the mesoderm. And the cells of the original epiblast become known as the ectoderm. Okay. So through this process of gastrulation, okay, we're forming these three primitive germ layers: the endoderm, the mesoderm, and the ectoderm. We're actually looking down on the surface of the embryo here. Let's go back. Above this surface of the embryo, here's the amniotic cavity. So this amniotic cavity is filling with fluid as the embryo is developing. And what's going to happen is this amniotic cavity is going to expand, and it's going to completely surround the developing embryo. So that during pregnancy, the body, the embryo, or later the fetus, is going to float within that fluid. Remember, this fluid is growing and expanding, and it's going to surround completely the developing embryo. Okay. So you can imagine, based upon the fact that the ectoderm is forming the surface of the embryo, that the ectoderm is going to give rise to skin, right? Because skin's what's on our surface. Okay. So this is a famous biologist who reported that it's not birth, marriage, or death, but gastrulation, the formation of that trilaminar disc, which is the most important time in your life. So during gastrulation,